Hello, everybody, and let's get started with why genetic variation is super mega awesome. All right, so first of all, life cycles of eukaryotic cells have an overall pattern, and it alternates between mitosis and meiosis. So mitosis, then meiosis, then back to mitosis, then meiosis, etc., and so on. Okay, and basically this happens because two haploid cells from different parents, one from a female parent, one from a male parent, for animals at least, organisms, so they fuse. So you take a sperm plus an egg, they fuse via a process called fertilization, and they create a zygote. Didn't save myself enough space there, which is diploid. And that cell has all new gene combinations that have never existed before. Okay. And then that zygote divides via mitosis to create a multicellular organism. That multicellular organism at some point undergoes meiosis to produce new haploid cells, new gametes, which fuse and become a zygote, and then divide via mitosis, and so on and so forth. Okay? All right. So we alternate between mitosis and meiosis. And this sexual reproduction is so important because it creates, again, our favorite new friend, genetic variability or genetic variation. This is essential for surviving in a changing environment. All right. So here's our good friend, evolution. Okay. Big idea one, super important. We're all just trying to make it to the next generation. And to do that, we need to be able to be flexible, to be, adaptable. And having variety of genes, a variety of different alleles is the best way we can adapt most easily to our environment. Okay, so there are several ways. I think I'm going to talk about 4 ways that we can get genetic variability. So the first way is by mutation. Okay, mutations produce new genes, new variations, but are relatively rare. And this is because your DNA has a lot of things that try to prevent mutations, because mutations, as we know, aren't always good mutations. Sometimes they're not so good. So it can create genetic variation, but it's not the only way. We can't depend on that in order to create enough genetic variation to survive. Okay? So the majority of genetic variation results from meiosis and sexual reproduction. Okay, so let's look at where it happens during those processes. So the first way that we get genetic variability during meiosis is the shuffling of homologs. So this is kind of genetic variation method number 2. Okay, and this is what we were talking about in the other screencast, independent assortment. And this is going to be a big deal when we move on to the next chapter, which is genetics. Okay, so super big deal there. All right, so when chromosomes, when the homologous pairs line up on the metaphase plate during metaphase 1 and separate, this increases genetic variation because you're putting different chromosomes, even though they're the same chromosome, they're both chromosome 1 or both chromosome 2, they're different versions of those chromosomes. So they have slightly different versions of the genes. Okay. And again, our law of independent assortment basically says that which of the 2 homologs from each pair end up in which daughter cell is completely random and doesn't depend on any other pair of chromosomes. Okay, so it's completely independent. And So the number of possible chromosome combinations, the way they can orient themselves and segregate into separate daughter cells, can be calculated by 2 to the n, where n is equal to the number of homologous pairs, so our haploid number. So if we do a little math, hooray, yay math. Humans have 23 pairs of chromosomes. So independent assortment can create gametes with 8 million possible combinations of chromosomes. Okay, so that's 2 to the 23rd. All right, so already we're up to, mutations can, create a couple of different alleles every once in a while. But every time the cell goes through meiosis, there's a possibility of 8 million different combinations of chromosomes in those 4 cells. Okay, so we're getting a little bit better. We're getting some better numbers here. So that's my second way. And this is kind of a little just diagram here. If we have, this is just 3 chromosome pairs. Okay, so if we have just 3 pairs, there are 4 possible arrangements of the chromosomes at the metaphase plate, and you can see them up here. And then the 8 possible sets of chromosomes after meiosis 1 is down here. And so you can see even just with a couple of different pairs of chromosomes, it increases exponentially, obviously, and we get lots of different arrangements of the chromosomes in our daughter cells. So that is the second way. So we've got mutation creating genetic variation. We've got independent assortment creating genetic variation. And of course, the big daddy, the mother lode. The third way that genetic variation is created is by crossing over and recombination. Okay, crossing over creates chromosomes with novel combinations of genes, all new chromosomes that have never existed before. Okay, sexual reproduction brings together the homologous chromosomes from very vastly diverse populations. Crossing over can introduce allelic combinations that are completely new, completely novel. And then resulting gametes that are produced from crossing over are, gold mines for evolution of the organism. Okay, crossing over, changing the combinations of genes, swapping genetic material, ensures that no pure maternal or paternal chromosomes exist within humans. We're all kind of blends of all of our ancestors, which is kind of neat. You'll see, some of you may have, some similarities to your grandmother, to your grandfather on either side, maybe to a great aunt. And that's because some of their DNA is still in you, which is kind of neat. All right, so that is way number 3. And then our fourth way, I was right, to generate genetic variability is fusion of gametes. And again, of all the gametes that exist, there are many, many, many gametes that come to the table, or that can potentially come to the table when you get a fertilization event. Okay, each of those will contain a unique combination of alleles, and then they fuse to form another completely unique individual. Okay, so each gamete, by independent assortment, can have 2 to the n possibilities. Fusion of gametes with 2 individuals can have 2 to the n times 2 to the n. So gametes from 2 humans, even without recombination, okay, even not taking recombination into account, just independent assortment can produce 64 trillion different combinations, or 64 trillion different individuals. So every time there's a fertilization event, 1 egg and 1 sperm, it's kind of 1 out of the 64 trillion possibilities that exist just with independent assortment alone. When you put recombination on top of that, it's pretty much infinite, the number of characteristics that we can have. Okay, so again, fusion of gametes taken together with independent assortment and crossing over assures that every human individual is absolutely genetically unique. Okay, there's not another you, there's just you. Enjoy being you. That's my message. Be you, because you're the only you that there is. Korvasek out.